Thanks so much, everyone, for watching. We've got an epic lineup tonight of speakers. We've got an awesome panel here. Amazing group of functional doctors, technology, activists, for a leap forward in medicine.
You know, our goal is to make it easy for practitioners to learn in community.
This ecosystem that is being set up by the meetup groups, we believe will be the future ecosystem of this evolved primary care network, and it all revolves around you.
Welcome to the Functional Forum. Thanks for joining us in the charge to accelerate the evolution of medicine. Please welcome your host, James Maskell.
Hello and welcome to the Functional Forum. We are back in Chicago for the Personalized Lifestyle Medicine Institute's Metabolic Medicine Conference. It is the 10th year, and this year we are getting into one of the hottest topics, the cross-section of metabolism and immunity. We're going to hear about the microbiome, we're going to hear about mitochondria, aging, and all types of chronic illnesses. It's going to be an incredible session. Enjoy. So when you're dealing with the nature of complex chronic illness, obviously it's all interconnected, and a big foundation is systems biology. And so when Dr. Bland puts together these conferences, you're going to hear on all these different areas of medicine and how they come together. And so today we're going to get into the mitochondria. We're going to hear from Dr. Rob Knight. We're also going to hear from Dr. Jon Kaiser, who gave a great talk on mitochondria as potentially the root cause of chronic disease and aging. And then we're also going to hear from Dr. Kristine Burke, who talked about her universal barrier thesis. We're also going to hear from other practitioners who were there, but there was so much other content that I'd love to share that we didn't get a chance to put into the show. But I really recommend that you come to these conferences because it is so rich. So in functional medicine, one of the underlying theses has always been that the environment drives more of your outcomes than your genes. And this has been controversial because there's been the deterministic theory of medicine that's been running for over 100 years. So what is the mechanism by which the environment modulates human function? And that is the microbiome. And Dr. Rob Knight is one of the leaders in that conversation, one of the leading researchers in the world. We've never had him on the Functional Forum. The Functional Forum was started and stimulated to start because of the transformational research that was coming out when it comes to microbes and understanding the way that complex chronic illness works. And so I'm super excited to have Dr. Rob Knight on the show. Check out this segment with him. All right, we are here with Dr. Rob Knight. He keynoted today. We're going to have some of the best bits from his talk a little bit later in the show. But Doc, you know, after years of doing this, a decade, it finally feels like the microbiome is having its moment.
Yeah, I couldn't agree more. It's finally getting just from fun stuff we know in the lab towards things you can apply for real in your daily life.
Absolutely. And look, I want to share my appreciation because I feel like this topic is finally showing what I think functional medicine has known for a long time. The environment is critical, and this is the interface.
Absolutely, I couldn't agree more, especially these themes about how important food is, how important environmental exposures are, how important the immune system is. The microbiome is one thing that really connects all of those, but then other things you're hearing about today, like the critical role of mitochondria, also very important. And when we can tie those together in an integrative context, we'll be able to help patients so much more than when we just view one thing at a time.
Awesome. Well, we got some of the best bits from Dr. Knight's lecture. Enjoy.
Great, thank you. So one of the main things that we're interested in at the moment is the gut-brain axis. And I'm going to talk for just a little about our large-scale project funded by the National Institute of Aging, the Alzheimer Gut Microbiome Project, where to a large extent we're piggybacking on already funded clinical trials of diet related to Alzheimer's, so MIND, BEAT-AD, and US Pointer. And you saw Jeff refer to one of these studies, the MMKD, the Modified Mediterranean Ketogenic Diet, in his talk just before. This just gives you an idea of the people and institutes involved. So it's a huge multidisciplinary collaboration, and I'm one of the leads of the project together with Sarkis Masmanian at Caltech and Rima Kadara-Dauk at Duke University. So one of the key successes of this project that was published in Nature Biotechnology a couple of years ago was this idea of reference data-driven metabolomics, where the idea is, what if we could do diet readout from clinical samples directly by looking at the molecules in those samples? And this actually came from an idea from the microbiome data in the Earth Microbiome Project, where a lot of the microbes, especially from environments like marine sediments, we can't identify them, but for a new sample we can say we don't know what these microbes are, but we know a lot of them come from one source or another. And so this was an application of that idea to molecules, where you can take your clinical samples and then you can take that food reference and say, okay, I don't know what this molecule is, but I know it came from a salmon or from an onion or from an egg or from some other dietary source. So what I'm going to do then is just count up all of the matches that I got to each of those sources, attribute them to each individual, and then ask, can I say anything about the molecules from these different dietary sources that I saw in that individual when I connect it to phenotype? So just for face validity, here's a PCA of the food counts, where you can see that the omnivores in blue and the vegetarians in brown separate really nicely in a principal components analysis of the food item inventories. Then if you look at the individual components, they're what you'd expect. So the omnivores eat more dairy and meat, the vegetarians eat more legumes and fruit and so forth. So having established face validity, the next part was to ask, well, what happens if we correlate with phenotype? And so we looked at Ray's auditory verbal learning test, which is a very standard measure of auditory learning that has 4 subscores. And so what we could do is we could basically take the spectra associated with each food item and then correlate it according to each of these subscores with what was good or bad performance on memory for each food item. And so what we saw is that the foods that were worst for memory, and this was consistent across subscores, were sugarcane and soda, whereas the ones that were best were fish, strawberries, and mushrooms. So you might be saying, well, so what? I could have picked any food out of a hat and it would have been approximately as convincing. But when we went back to the literature and the popular press accounts, there's a giant literature about exactly how bad soda is for your memory, especially with specific reference to Alzheimer's. And then similarly, there's a huge literature on how bad sugar is for your memory, including specifically for Alzheimer's. So much so that Alan Saltiel, who leads our Diabetes Research Center at UC San Diego, often says that he thinks that Alzheimer's should be considered a third form of diabetes, along with type 1 and type 2. We should be calling Alzheimer's type 3 diabetes. And then at the other end of the scale, there's a giant literature on how good fish is for your memory, again, specifically in the context of dementia and in the context of Alzheimer's. And also both for strawberries and for mushrooms, we found a few specific studies that demonstrate, again in the specific context of Alzheimer's, that both of those reduce your risk of developing dementia. And so this is really exciting because essentially what we did is we replaced thousands of those heroic Lewis and Clark style expeditions looking at individual food items in different populations with basically an afternoon of work on the mass spec. And so the potential to do this for anything, not just for memory, but for other physiological scores, for any phenotype, not just for gut-brain axis related ones, but if you want to look at metabolic phenotypes, immunological phenotypes, you can absolutely do it with this kind of technology, relating food items to what you care about. Okay, so having established these links between food and the microbiome and health, that leads to the really obvious question of how much can you change your microbiome? And at that first meeting that Jeff referenced of PLMI, I was really taken by a quote from Jeff's talk where he said that food is a language that speaks to our genes. So think about that for a moment. What he's talking about there is that our genes are fixed at conception, but what they do depends tremendously on what we eat. And there's generic advice that's very good advice, but doesn't work for everyone, that you should eat the rainbow, right? And so what that's speaking to is that this language of food is very much a language of color, but that language of color is itself a language of chemistry. So everything from the carotenoids that make the carrots orange to the anthocyanins that make the berries purple, all of those are particular kinds of chemicals that we can read out from the mass spec, and that we're increasingly understanding how they intersect with all of those phenotypes. Now, regrettably though, even though I live in California, this is the source of food that's closest to my house. And when my daughter goes there, she certainly sees a lot of bright colors. And what I think is most ironic is their motto, "Too much good stuff," because sometimes I wonder whether all of the good stuff has been cynically stripped out of these products and replaced with synthetic analogs that have completely different physiological properties. And I'm not the only one concerned about this. So the AMA published this article a couple of years ago, What Doctors Wish Patients Knew About Ultra-Processed Foods. And I'm guessing you probably all know this or you wouldn't be here, but the article's just full of quotes you don't want to see, like the health consequences are dire. The concerns include recent documentation of risk of type 2 diabetes, cardiovascular disease, and dementia. But fortunately, if you've been keeping up with the popular press, you know there's an easy solution, because at about the same time this article came out, cheese protects you from all causes of death, says science. And I mean, why would science say it if it weren't true, right? It's just such wonderful news. Finally, humanity's battle with the Grim Reaper is over. All we had to do was find exactly the right piece of cheese to fight him off with. But in all seriousness, this kind of nutrition research is really hard. And this paper from Daresh Mozaffarian and his collaborators that came out in the New England Journal back in 2011 is still one of the largest scale efforts of its kind. And what they were looking at was changes in diet and lifestyle and long-term weight gain in women and men. So they were looking at every food item and the impact on weight loss and weight gain. And again, you could say that the results were startlingly obvious, because if you look at the foods that make people that are associated with weight loss or weight gain, they were exactly what I would have expected from stereotypes, right? So if you look at the food most associated with weight loss, it's yogurt. Think about all those commercials featuring thin women laughing at their cup of yogurt. And then on the other side, the food most associated with weight gain was that friend of every pediatrician's office, the potato chip, right? So that was maybe not very surprising, but what was surprising to me was the size of the effect. So each serving of yogurt per day led to a weight loss of 372 grams over each 4-year period in this 20-year study. Whereas in contrast, each serving of potato chips per day led to a weight gain of 768 grams over that same 4-year period. So in other words, if you were going to take the results of the study and tell people to give up the worst food that they were eating a serving of every day, substitute it with the best food, and making that a good choice every single time, 1,460 days in a row, all it's going to do is change your weight by a couple of pounds, which is pretty depressing, right? But what this is masking is that these are population average results, but everyone is different. And as I mentioned, a lot of those differences are traceable to the microbiome. So this is about physical characteristics, but there's also a tremendous and not that well recognized link between diet and mental health. So the Food and Mood Center at Deakin University conducted the SMILES trial about 7 years ago now, where what they were looking at was they were looking at diet therapy for individuals who were newly diagnosed with major depressive disorder. And what they found was that on the Mediterranean diet, about 32% of them recovered, which is about equivalent to what you would get with SSRIs, which is standard of care for MDD. And as a control, to make sure it wasn't contact with the research team and the counseling and so on, they put a control group on a low-fat diet where only 8% of them recovered. So that's how they could demonstrate that it was due to the specific diet, not just due to counseling about diet in general. And they followed this up with their Healthy Med study, where they were comparing Mediterranean diet to social group therapy, which is standard of care non-pharmaceutical intervention, and also about 30% effective. And what you can see is, again, the Mediterranean diet did about as well as standard of care therapy. And there's increasing support for this. So, for example, a couple of years ago, this meta-analysis came out on diet and depression. So it's a systematic review of whole dietary interventions as treatment in patients with depression, which essentially concluded that there was some evidence supporting the Mediterranean diet. But I have to say that reading this paper made me somewhat depressed, because they had to read 1,863 studies, and only 5 of those 1,863 met their quality criteria for inclusion. So it's not that we need more research into this stuff, we need more high quality research that you're able to draw adequate conclusions from, with reasonable sample sizes, with randomization, and with lack of methodological bias, all of which are huge problems in the literature at the moment. And I'm sure you all know about the field of nutrigenomics, which has been looking in the human genome for the key that unlocks these relationships between diet and health. And searching for this key has cost billions of dollars already. But I start to think, especially given the lack of success of these nutrigenomic tests, that we should really be looking elsewhere, such as in the microbiome. And a large part of the reason why we should look in the microbiome is it's just such a larger source of genetic variability. So each of us has about 20,000 human genes, but we have from 2 to 20 million microbial genes. And so what that means is that you could say at the level of individual genes, we're at best 1% human, and we're ignoring 99% of them when we look only at the human genome. But what's more important is that we're ignoring the 99% that we can change through things like diet and drugs, rather than by gene therapy.
All right, I'm here with Kristine. She's learning along with the rest of the crew. Kristine, what did you find interesting this morning?
I mean, the entire conference has been beyond interesting, but most interesting, I think, has been the focus on the mitochondria and its role and what a powerhouse it really is. I think it's just so overlooked in medicine. I've been in medicine for 20 plus years, and never until this conference have I heard such emphasis on the importance of the mitochondrial cell.
Do you feel empowered to go and do something about it in the patients that you're seeing?
I mean, I feel empowered to educate my patients about it so that they can feel empowered to tackle their health on a totally different level. I mean, functional health is something that I was never a part of. I didn't practice in functional health. I practiced in a hospital environment for 20 years, and I've been 1 year in functional health, and I am just mesmerized by the knowledge that functional health brings to medicine and the health of the human body as a whole.
I'd love for you to just share like how it's different being in those two worlds and how you feel about your work.
It's fascinatingly different. I mean, really the biggest difference is I think functional medicine puts a lot more power into the patient's hands to tackle their own health and understand why they're unwell, where traditional medicine, hospital medicine, has put the heavy focus on a physician just sitting down and telling a patient what's wrong with them, even though the patient knows their body the most. The physician certainly doesn't. And it's a complete paradigm shift that I'm fortunate to be a part of.
Well, I'm glad you're here. Enjoy the rest of the conference.
Thanks so much. Appreciate it.
Cheers.
Thank
Enjoy.
you. Yeah.
All right, I'm here with Dr. Tom Williams. Doc, you know, when Dr. Bland puts these events together, this is on immunometabolism, but you're charged with talking about the liver. Why is that a critical part of this conversation?
Well, the liver is, you know, what we always thought is the key metabolic organ in the body, and it's really tasked with doing so much. And our connection between the immune system, and we're actually going to be talking about the microbiome. I'm going to be talking about the gut-liver and the gut-brain, and actually now we know as the gut-liver-brain axis. So these metabolic signals that come out of the gut, many of them make it through with the portal vein into the liver. The liver's really tasked to sort of coordinate what is going to go out to the rest of the body, what signals. We think of it, you know, the cholesterol packaging, you know, lipid metabolism, all kinds of, you know, detoxification. And so there's a robust relationship between how the liver manages its metabolism and really how the gut and the gut microbiome metabolize, and there's a lot of crosstalk. So my talk is going to be a little bit about how, you know, the new nomenclature between the fatty liver disease. And we no longer call it fatty liver disease, but we're going to talk about it as a metabolic disease. And we're going to see that all the things that we've been talking about, the mitochondria, the gut, and all these other signals are all kind of flowing through, and the liver is kind of managing that.
What did you think about Rob Knight's talk this morning?
So, I mean, I think, well, it was great because he's at the heart of the history of understanding the microbiome modulation, and not just in humans, but he talked about, the microbiomes and all these ecosystems, and understanding, sort of that, the history of understanding how much the technology that we have gained in the last 15 years has radically altered the amount of information we have. And slowly but surely, we're in with AI and with other models, trying to make sense of what do we know and how can we alter that for the clinician, because you have these huge labs and all these data sets, but then the question is, how does a physician take the information they can gain from the patient that walks in the office, give them some sort of protocol, and then see an outcome? And I just was talking to Rob just before this interview, and that still is the challenge. How do you take all that data and make it into a clinical decision-making process?
It does seem that even though the data gets more complicated, eventually the solutions end up being reasonably simple in some cases.
Yeah, I think for the most part, we're still dealing with diet and kind of lifestyle factors, because the microbiome is one of the great interpreters of the environment for us. But I think we're learning also that certain people don't respond to what we thought they were going to respond. So 80% of the people probably respond a certain way, and then those 20% of the people maybe have anomalous response, no response, or maybe an exaggerated response that's opposite of what you'd expect. And so the question is, can we predict that? And can we direct them towards, because those are the people that probably go from physician to physician to physician to physician, and nobody's figured out that 20%. And those are the 20% of the complicated patients. Unfortunately, the 80% of the people that aren't even going to clinicians and don't care about their lifestyle, or aren't making the connection, or are unwilling to change their lifestyle, those are the people that we'd like to be able to change.
Yeah. You're speaking tomorrow, but you come to the whole weekend, and ultimately, how is it like lecturing after you've taken in 7 hours of some new stuff you've heard before, and then try to put it into something where you've already given away your slides?
Well, so my slides were sent in, I don't know, 6 weeks ago or whatever it was. And now you'd like to go back and can I change all these slides or rearrange them, or somebody already talked about this part of it. But that's, I think, as I've learned, and I think many clinicians learn, to hear it a second or even a third time. But I usually say it maybe from a different angle, and it takes a while to click in and make aha moments. And some of these clinicians, they have parts and pieces. So maybe I'm only going to speak. Only maybe 5 slides in my talk might be relevant to one person based on the knowledge that they have. So I always like to engage, and hopefully I'm going to add to the knowledge base that people come out with.
Well, I'm sure you will. We're looking forward to it. Thanks, Tom.
Good seeing you.
Cheers. All right, I'm here at PLMI with Dr. Jon Kaiser. Doc, the last time we spoke, you were talking about mitochondrial health, and I know that's a passion and through line for your career, but we were talking about it in the context of post-viral illness and long COVID. This time at this conference, you were talking about it in relation to aging and chronic illness. Can you share sort of your journey in that, how you got from one to the other?
Yeah, well, so the mitochondria support the health of the cell. They make all the energy. They control whether it lives or dies. And in the context of this conference, we've been seeing this epidemic rise in chronic disease. Autoimmune disease, cancer, neurodegenerative disease, Alzheimer's disease, and the current theory of medicine, the germ theory, where there's one target that you aim a toxic molecule to, an antibiotic or a blocker. It just doesn't work when you're treating a complex chronic disease. And the new theory that I've been drawn to is called the mitochondrial cascade theory of aging and disease. And what it holds that we may have healthy mitochondria, in our early years, in our 20s, in our 30s, but over the course of our lifetimes, we're exposed to toxins, chronic stress, excess alcohol, pollution, and viral infections that damage our mitochondria and degrade its ability to function. And as the cells of the body have less and less energy to make use of, there comes a critical point that your genetic weak link can no longer hold things together. And if it's in the brain, you start developing Alzheimer's or Parkinson's disease. If it's in the liver, you start getting liver dysfunction. And autoimmune diseases if it's in the immune system. And what they've traced these diseases to as the trigger is a serious decline in mitochondrial function. So I believe everybody should be supporting the health of their mitochondria with a range of antioxidants, starting with a good, healthy plant-based diet, the rainbow diet, if you will, intelligent evidence-based use of supplements. But when you start to get the symptoms of these diseases, the earlier you intervene at supporting and reviving your mitochondria or repairing your mitochondria, because they can repair, they reproduce, they turn over, then you can turn these diseases around before they become devastating.
One of the interesting things you had in your slides were that if you target the mitochondria with therapeutics, then it helps everything else work better. What do you think is the mechanism for that, and what are some examples that you could give of where you've seen supporting mitochondria and it helping something else be more effective?
So the mitochondria, we know from electron microscopy that in cells that are struggling to function, that the mitochondria are damaged. You can see damaged mitochondria. And we know that by giving certain combinations of antioxidants that we can help the pool of mitochondria repair itself, and we can elevate the number of intact functioning mitochondria. And so if the cells of an organ don't have enough energy, then they go into what's called apoptosis, which is a programmed cell death, and they shut down. So if you help the mitochondria repair and you increase the amount of healthy energy available to the cell, you avoid going down that apoptosis path, and you preserve the health of not only the cell, but the organs.
So you're talking broadly about chronic illness, but then ultimately you had a plan, and the plan was a new study that you felt like could open up everything. And out of all the things that you could look at, you thought that mild cognitive impairment was the place to start to get the data that you think is needed to shift the paradigm. Right. And I just love to help the people at home understand why you've come to that conclusion.
Yeah. So back to this shift from the germ theory to the mitochondrial dysfunction theory. There is an abundance of research evidence that points to decline in mitochondrial function as, if not the trigger, the cause of Alzheimer's disease and Parkinson's disease. Let's just take neurodegenerative disease. And if that is true, we need to intervene to revive the health of the mitochondria, but we also need to, look at this broadly, because everything affects the health of the mitochondria. If you're exposed to toxins, the food you eat, the amount of stress you're under, things like that. So what we need to do is we need to take a functional medicine approach to these diseases, right? And so, like I said, there's plenty of evidence supporting this theory, mitochondrial cascade theory, but what is missing is a pivotal study, a study that's going to show that head-to-head, if you take two groups of patients with early Alzheimer's called mild cognitive impairment, you want to do an earlier version of the disease, because that's when you can get the most reversal.
Yep.
So if you take two matched groups with early Alzheimer's disease, and you apply functional medicine paradigm to treating one of the groups, and you apply standard conventional care to treating the others. If you take a look at the effect on cognitive scores, the effect on biomarkers like p-tau217, and the effect on imaging, the size of the frontal cortex and the hippocampus, and you follow these patients over a year, but you also have to not only look at these outcome measures that I just mentioned, you need to look at the overall cost of care. So my thesis is that in a pivotal study, if you go head-to-head functional medicine treatment versus conventional care in early Alzheimer's, if you show improved outcomes and decreased cost of care, you're going to get the attention of the payers, because the payers are looking at a doubling of dementia cases over the next 25 years, and they are wondering how they're going to avoid bankruptcy. And so you've money, the world, money drives a lot. And so if you can show decreased costs of care and improved outcomes, then you're going to get the interest of third-party payers and the federal government to, if not move to supporting the treatment with functional care, at least funding additional trials, and then you start to see a shift in the paradigm. But you need to have both improved efficacy and decreased cost of care. And from the data I've seen, Dr. Bredeson's case studies and the work that Kristine Burke is doing, I have a high level of confidence that that study would be overwhelmingly successful, and that's how you shift the paradigm.
Amazing. One of the root causes that you spoke of, mitochondrial dysfunction yesterday, was microplastics, and amongst all of those other ones, all the other ones that you mentioned, we sort of have good ways of measuring, and it's been around in functional medicine for a while. I mean, I agree with you, and I would just say, now that you're aware of this, how do you bring your patients into that conversation, and how do you look to help them understand where their unique risk is for that topic?
Right. So this speaks to the increasing amount of plastic in our environment, and what people don't realize is that plastic breaks down and breaks down and breaks down into these microplastic and nanoplastic shards. The people who are doing the research have used this term nanoplastic shards because the breakdown products are very tiny, they have rigid, sharp edges, and they damage sensitive structures. They get embedded in the membranes of nerve cells, brain cells, and mitochondria. And so we know that this is happening. We need to definitely do more studies on it, but everybody should be trying to minimize their use and exposure to plastics in their environment. Plastics are in our clothes, so buy fewer clothes with polyester and latex. Plastic is in our kitchens, so avoid buying plastic wrap and cooking your food in plastic containers in the microwave. Microplastics are in the dust in our house, so we need to have air purifiers. But we need to start pushing back on businesses and companies to use less plastic in their packaging. We need everybody to start where they are and do what they can to minimize plastic in their environment, because there is not a blood test, there is not an X-ray, there is not a CAT scan that shows this up. But what the researchers are finding is the closer a person is to the present moment, the higher the concentration of plastic in their body. And these microplastics are concentrated in the brain, not in the liver, not in the kidneys. There's 7 to 30 times more microplastic fragments in our brains than in any other part of our body. It's worrisome. It's something everybody should become aware of, and we need to do a better and better job.
Well, Doc, I really appreciated your session yesterday, and I really think this has been a constant theme throughout the Functional Forum. This concept of the importance of the mitochondria, and I thank you for making it so clear and delivering such a great message. We're gonna have some of the best bits from Dr. Kaiser's session. Enjoy.
Microplastics preferentially concentrate in the brain over any other tissue of the body. They thought they would concentrate in the liver, right? The liver's trying to clean them out, or the kidneys. There are 7 to 30 times more microplastics in your frontal cortex than in any other tissue of the body, and they drive toxic outcomes and exacerbate disease. Now, this paper went a step further. Microplastics are plastic particles you can't see. Nanoplastics are even smaller. Now, plastics are polymers, right? Polyester, polyurethane, they're polymers. And they progressively break down into smaller and smaller pieces. And they use the term, and it's not an accident, shard-like fragments. So I decided to look, what is a shard? A shard is something with rigid, sharp edges capable of doing damage. That's the dictionary definition. Rigid, sharp edges capable of doing damage. So on the left is the old resolution, and on the right is the new resolution, and you can see they almost look like little pieces of glass embedded in the membranes of brain cells. So they decided to plot the concentrations from 3 different cohorts, and these are from biobanks. And so they looked at brains from the early 2000s, the concentration of micro and nanoplastics in the frontal cortex. And then they looked at a cohort from 2017, and then they looked at a cohort from 2023. And as you can see, it's a straight line increase in the concentration of microplastics in our brains. What they found was about 2 grams is the average concentration of plastic in each frontal cortex. That's a heaping teaspoon of plastic particles. Now, these are normal brains, right? These patients had normal brains. When they took a look at the brains of patients with dementia, orders of magnitude higher concentrations of plastic. So this is my rendition of what a nanoplastic shard looks like when it's embedded in one of your membranes. I didn't do this on purpose. This was a garden accident that occurred while I was putting this talk together. Just serendipitously, it occurred in my garden, and I looked at it and I said, oh my God, that's a plastic shard in a membrane. And you can see how it can do very significant damage to these sensitive mitochondrial membranes and neuronal membranes. And the problem, nobody's putting the brakes on. That's the scary part. Nobody's putting the brakes on. And we need to, because we're headed towards 1 billion pounds of plastic a year being released into the environment. Every animal, every penguin in Antarctica, every mountaintop we found microplastics. So this is a very serious problem.
All right, we're here with Cassandra Haddad, practitioner at the Turnpole Clinic. Learn anything interesting this morning?
Yeah, it was a great morning packed with wonderful information, but I think one of my big takeaways was that diet is just as effective as SSRIs, which are also known as antidepressants, or group counseling. So diet, back to basics, super important.
So you could think that, imagine doing those things together, right? Diet, having a group connected, like that seems like that's gonna outperform everything.
Absolutely. I think medication was never meant to work by itself. We've known that for a really long time. It's a tool that can be used in the toolbox, but we can't forget about the other tools like diet and group therapy and individual therapy.
Awesome. Well, thanks so much for sharing and enjoy the rest of the conference.
Absolutely, thanks.
As you've already seen, there's a lot of heady science, as ever, in the PLMI. Ultimately, what I think most practitioners are looking for is a structure by which they can think about how to take action and work with patients in a way that is consistently successful. And one of the most intriguing parts was Dr. Burke's unified barrier hypothesis. If you've been in functional medicine for a long time, you're very familiar with the intestinal permeability as a foundational concept in functional medicine. But what are the other barriers in the body, and how can we think of it as one barrier, as a leverage point for reversing complex chronic illness? Enjoy.
So here's where we are. I think that the ultimate key to our defense in this process, how we protect ourselves, is our barriers. And so today I'm going to present to you what I'm calling the unified barrier hypothesis. So we all know that the vasculature is super foundational for transport, for nutrients, for oxygenation, but it also is a key regulator of overall homeostasis and health. And when you look at the diagram, or not the diagram, but the image of the vessels from the body's exhibit, you can see how densely concentrated the arterial supply is in the brain and all around the mouth. Let's do a little blood-brain barrier refresher, because if you're like me, that was a little bit in the dusty recesses of my med school days. So first of all, we know it's a highly selective membrane, and we also know that it's localized in the central nervous system vessels, all of the central nervous system vessels essentially, and it's structurally unique with the astrocytes and the pericytes surrounding it. So the astrocytes in blue are the ones that cover nearly every millimeter of surface in the cerebral vasculature, and they provide basement membrane components. They release signaling molecules like TGF-beta, which initially is a calming cytokine, but then when it gets elevated into the tens of thousands is highly pro-inflammatory. They have a role in maintaining the tight junctions of the blood-brain barrier, and then they also provide immune surveillance, and they modulate inflammation and help to repair injury. The pericytes have a little bit more structural role. They help to maintain the tight junctions as well. They orchestrate the endothelial cell activity using cytokines, and then they also are going to help facilitate the removal of amyloid from the brain. So really important role there. Then we have our two transporters, which I can kind of point at. Anyway, here on the left side, we've got the solute carrier transporters. So these are passive transporters. They use mostly gradient. They're gradient driven to move nutrients, to move glucose, for example. This is why the brain cannot control the amount of glucose that it contains. So if the blood sugar is elevated, it's a passive gradient driven transport. The brain glucose is elevated, and that leads to brain insulin resistance as well. Then we have the ATP binding cassette transporter proteins. These are active pumps, so they are ATP dependent. They limit accumulation of things like xenobiotics, drugs, toxins, also in conjunction with the P-glycoprotein pumps. Those are the ones that keep the lipid-soluble medications out of the brain. And so these are highly dependent on the energy capacity, i.e. the mitochondrial function of the brain tissue. Then on the other side of the blood vessel, we know that the endothelium is the real workhorse of the vascular system. It has a critical role in a number of different things that we'll touch on here in a moment. We know that the endothelial glycocalyx, which is that slimy inner lining inside the endothelial cells, projecting into the lumen of the vessel. So that is microscopically thin. It's got a gel-like consistency, and it creates basically a non-stick layer. And it's made of the glycoproteins, the proteoglycans, and the glycosaminoglycans. And that's important because those are all building blocks that we can use to support the production of glycocalyx. It's both a physical barrier because it's a dense mesh, and it's also an electrical barrier because it's highly negatively charged. Do we go? Yep, and then it thins with age, and then with all of the different inputs that create inflammation, those will all also damage the endothelial glycocalyx. All right. Oh, that didn't click. So this is what it looks like under electron microscopy, and with electron microscopy and the improvements in digital image resolution, we can now see these fibers projecting into the arterial lumen. So what does the EGX do? All of the things that we used to think the endothelium did. So it's the gatekeeper. It creates the selective permeability. A healthy glycocalyx is a dense mat, and so it's able to harbor the antioxidant enzymes like extracellular superoxide dismutase. And it also transmits the shear stress of the wave action, so kind of like kelp, right? As the blood is pumped, the endothelial glycocalyx waves in that blood flow, and that stimulates the production of nitric oxide. But when the glycocalyx is unhealthy, like we see in the bottom panel, then we have turbulence to the blood flow, we have loss of that protection for the permeability, and we see increased vascular permeability, and then we also see increased adhesion of the leukocytes and then translocation of the leukocytes into the endothelium. That's how plaque forms in the arteries. So how is the glycocalyx damaged? So I mentioned that all manner of causes of inflammation are going to contribute to that, but it's really going to be modulated through the production of inflammatory cytokines that trigger this enzyme class called shedases. And shedases are enzymes that cleave the glycocalyx at the level of the membrane. So I like to think of it like a lawnmower. So it comes along, it shaves off the glycocalyx, it leaves the intramembrane component so it can be rebuilt from that, just like the grass will regrow. But it cleaves off the part that's providing that biochemical structure. Also, reactive oxygen species, so oxidative stress, will damage the glycocalyx. Hyperglycemia is one of the most powerful damagers of the glycocalyx, and that makes sense when you think about, that's right, that makes sense when you think about how much greater risk diabetics have for cardiovascular disease than the average population. So half of us, the normal population, die from cardiovascular disease, but 2/3 of diabetics do. Diabetics are also twice as likely to get dementia. That was brought up today with the proposal to call it type 3 diabetes. And endotoxins and infections. So all of the things that we know about that will create inflammation. In the interest of time, I'm going to skip that one because I got better cool things to talk about. So this is really interesting. So they were looking at the density, the volume of endothelial glycocalyx in various tissue types, in various organs. So in A, we have the cerebrum, B, we have cardiac, and C, we have pulmonary. And then in the graph, you can see that the glycocalyx in the brain is 2 to 4 times denser than it is in the periphery. So it has a really, really key role in that. That makes the endothelial glycocalyx a key strategic inflection point if we want to look at what do we need to maintain, what do we need to protect, how do we need to keep the blood-brain barrier integrity intact, it's by focusing on the glycocalyx. So we have another barrier that we're all very familiar with that's critically important, and that's our gut barrier. So by a show of hands, does anybody know exactly what this structure is that I'm showing? All right. You're my first taker ever. So this is the intestinal glycocalyx. Isn't finding new things in the body really fascinating? So this has been there all along, obviously, but we haven't been able to visualize it because all of our electron microscopy techniques up until now use desiccation. And this is a very, very fragile structure, and so it's been collapsed and just looked like a bit of fuzz on the microvilli. But it's really a very, very robust structure, and arguably probably one of the most important physiologic structures for this purpose. So now our intestinal barrier system becomes so much more than a single cell layer of epithelium that we have to maintain, because it's really a complex 4-layer barrier system. We have the mucus layer in the colon, we have a very dense inner mucus layer that prevents adhesion of the microbes, because of course the colon is where our microbiome is housed. Then we have the glycocalyx, and then we have the intestinal epithelium. So there's a lot of barriers, which really in the body, that just tells you how important the barrier system is, because we have multiple layers of protection to try to keep it from being breached. So this slide really lays out the story of how gut problems turn into brain problems. So on the left-hand side, in a healthy gut, we have a nice, dense mucus layer and intestinal glycocalyx. We have intact epithelium, and all of the things that are supposed to stay in the gut, like LPS, lipopolysaccharide, our microbes, amyloid, are all in the gut. And then we have this nice balance of immune conversation with the immune cells, the astrocytes and the microglia, and through the enteric nervous system there in the middle, our HPA axis is balanced, and then our cytokines, our neurotransmitters are being circulated from the gut up to the brain. Everything is happy. But on the right-hand side, that's where we see what happens with a leaky gut and how leaky gut directly leads to neurodegeneration. So we have the loss of the structural barrier. So now we have LPS coming in, triggering an immune response. We have cytokines and bacterial metabolites leading to a disruption of the blood-brain barrier, which is the little pink cells along the middle of the slide. We have that blood-brain barrier disruption now leading to activation of the microglia. That leads directly to our neuroinflammation and neurodegeneration. So you cannot have a healthy brain without a healthy gut. They're inexorably linked. So what does all this mean in the context of the pathology that we often see in Alzheimer's disease? And I say often because I actually even had this conversation earlier today. We see people late in life, in their 80s, 90s, with robust amounts of amyloid who did not have cognitive decline at autopsy, obviously. And then we see people that have symptoms consistent with an Alzheimer's type presentation who don't end up having a lot of plaque. So it's a piece of the puzzle. It's a strong contributor, but it isn't an if this, then that. As always, there's more complexity to the puzzle. So let's look at how it unravels. So in the middle, we have our common pathways of chronic disease. We have chronic inflammation, oxidative stress, cytokine imbalance, and we see those commonly lead to things like diabetes and to rheumatoid arthritis or inflammatory bowel disease. And in the brain, we see that lead to things like amyloid plaque, tau tangles, and then the cognitive decline and neuroinflammation pathways. So how is amyloid beta actually formed? What is it that triggers that process? So on the left side, we have in the healthy state, Normally, we have amyloid precursor protein, which is a transmembrane protein, and that middle transmembrane section, the red section, that's the amyloid beta peptide. So in a healthy situation, alpha secretase cleaves the APP at the at the middle, so that the amyloid beta peptide is divided and that doesn't clump together. But in situations of either the inherited ones like the APP or the p-senilin mutations, or in the setting of chronic inflammation, beta and gamma secretase get upregulated and they cleave amyloid precursor protein at the membrane level and leave that amyloid beta fragment to then clump together, and that leads to the oligomers that lead to the amyloid beta plaques. So that's how inflammation is related to the formation of amyloid, just one way. Then outside the neuron, we have the amyloid beta cascade leading to those plaques that then accumulate and interfere with the neuronal synaptic signaling. And inside the cell, the amyloid beta stimulates the hyperphosphorylation of tau, so excessive phosphorylation of tau, and that stimulates the formation of the tau tangles. So that's how those things are happening. And when you look at it this way, the tau tangles, the amyloid plaque, the hot microglia, and the toxic astrocytes, those are the pathological characteristics that we see. But what really leads to the activation and the creation of all those things are the things that we're used to dealing with, like inflammation and infections or dysbiosis, toxins, some genetic components. So all of those pieces are what are contributing to the environment that's creating the pathology that we want to change.
All right, we're here with Dr. Chris Turnpour. Doc, you enjoying yourself and learning anything interesting?
Having a great time, lots of great information, really top-level science.
What was some clinical pearls you'll be taking away?
I thought a fascinating clinical pearl was the conversation on the microbiome, how the microbiome is 90% predictive of obesity, and basically your genes in your own DNA is only about 55% predictive of obesity. So it's really the microbiome that's driving obesity, and then the sequelae of how do we modulate the microbiome is really what we need to focus on.
Awesome. So you're going to be talking a little bit about adolescent obesity. Why is the adolescent the time to intervene?
So with adolescent obesity, what happens is we make changes to our brain, we make changes to all our systems, but very important that we make changes to our brain, and our brain is going through development. It's where our synaptic pruning is going on, our synaptogenesis is going on, and if we don't get to it if we don't get to it before it's fully developed, it kind of leads to semi-permanent changes for the rest of your life. So it sets you up for failure for life if you don't get to it early. So if you develop these things after your brain is fully developed, it can basically go back to the way it normally developed. But if you're neuroatypically developing as an adolescent, well, then you're setting the brick and mortar foundation for your brain function for the rest of your life.
Wow. Well, we're really looking forward to your lecture. Check out Turnpour Health, one of the coolest clinics in the country. Doc, good luck this afternoon.
Thank you.
Cheers.
Appreciate it.
Thank you so much for joining us here at the Functional Forum. I hope you can take this information and apply it to your clinical practice. Next month, we'll be coming from Estes Park, Colorado, as part of the Inspired IM conference. We'll see you there.